Let's move to the next topic, intragenic interaction. Intragenic interaction is otherwise called as allelic interaction. In the previous class, we discussed the intragenic interactions with one example, that is incomplete dominance. Let's see today what are the other examples of gene interaction. Gene interaction which is classified into two different types, that is intragenic as well as intergenic. Under intragenic, there are incomplete dominance, codominance and lethal as well as pleiotropy. Yesterday, previous class, we studied only one example, that is incomplete dominance. Now, we are going to discuss few other examples also. Let's see once again, let's review what you learned under intragenic interaction. What's the other name of intragenic interaction? Allelic interaction. So, this incomplete dominance, what's the example and gives the ratio? The example of incomplete dominance is color of the flower in Mirabilis jalapa. Mirabilis jalapa which produces 1:2:1 ratio. Another example is snapdragon. Snapdragon is called as antirrhinum, which also produces 1:2:1. Another example of this is codominance. The example is ABO blood group. Let's see, before learning other examples, let's see the molecular explanation of this incomplete dominance. Once again we review what are the characters of this, how are we going to interpret the lack of dominance. Here in F1 generation there is lack of dominance. Here what's the character? Red and white. But in F1 generation, normally which has to produce red color, it has to dominate. The homozygous dominant character parent will express the phenotypically. But in incomplete dominance, as the name indicates, incomplete dominance, there is only a 50% will express, so that it do not give red. What's the reason we are going to learn? So, what's the question? How are we going to interpret the lack of dominance and give explanation to the intermediate heterozygote phenotype, which produces a heterozygote phenotype. The heterozygote phenotype is incomplete. So, let's see once again. Here the parent is R1 into R2 R2. So, this parent is, this is homozygous dominant, which is red color, and this is homozygous recessive white. In the normal hybridization experiment in F1 generation, it must give the dominant character has to express. So, it should produce red. But in this case, in incomplete dominance, instead of red color, which will not produce the red, but it gives an intermediate color. So, this R1 and this R2 which shows pink color. So, what's the reason this pink color develops? So, that's the question. How are we going to interpret the lack of dominance and give explanation to the intermediate heterozygote phenotype? Let's see this R1. Here two homozygous dominant and recessive characters are there. Homozygous dominant is R1 which is red color. So, in this allele, so in the allele R1 which consists of an enzyme which synthesize the red color pigment. So, it gives the color red. So, there is an enzyme present in this R1. The functioning enzyme which produces or synthesize the red pigment, red color, so that the homozygous dominant gives the color red. In this case R2, it's the defective form. In the defective form or in the mutation form and the defective of nature, here it do not synthesize this enzyme R2 allele, which do not synthesize the functioning enzyme which produces red. So, lack of producing this red color pigment, so the flower color is white. Then what's the reason? It gives R1, R2 as pink color instead of red, it gives pink. So what's the reason? Here R1 and R2. So half of this homozygous dominant red is present. So this half number, that is 50%, is not sufficient to produce the red color. So it forms as a pink. This is the reason, the molecular explanation of the intermediate color pink will be produced during this cross. So this incomplete dominance, incompletely the dominant character expressed, so that it's known as incomplete dominance. So today the first question you are going to learn, how are we going to interpret the lack of dominance and give explanation to the intermediate color of heterozygote phenotype. These two are homozygous, this is heterozygous phenotype. So it gives the ratio 1:2:1 ratio. In the same example, we can find it in Snapdragon, that is otherwise called as anti-rhinum. In Snapdragon also the same ratio 1:2:1 ratio. Let's see what are the other examples of this. The intragenic interaction, codominance. There is the example called codominance. So in this case, codominance, these characters, the genotypic and the phenotypic characters are simultaneously expressed. It's the character of simultaneous expression of the genes. The genes express simultaneously. There are few examples are found in this. In human being, there is a ABO blood group. In ABO blood group, here IA and IB. So in the I gene, the IA and IB express together simultaneously, which expresses in the ratio 1:2:1 ratio. In plants, how it express? So there are few examples we can find it in codominance in plants. Red and white color flower. There is one of the examples, red color and white color flowers produced in camellia. Camellia is one example, and sickle cell anemia, that is sickle cell hemoglobin, another example. So here in plants, this codominance is very well noticed by the method of electrophoresis and this chromatography. Electrophoresis and chromatography. There is an example, let us see, in Gossypium hirsutum and Gossypium strutianum. Strutianum and Gossypium hirsutum, cotton, common cotton plant. Here in this cotton, Gossypium hirsutum, Gossypium hirsutum and Gossypium strutianum. These two examples are cotton, Gossypium hirsutum, Gossypium strutianum. In this, when the proteins, seed protein is undergo electrophoresis, it produces the bands under this chromatography. It produces the parental character which gives bands. When it is noticed in the hybrid of this Gossypium, the hybrid plants which shows different bands both derived from the parents, homozygous and heterozygous parents. So all these characters are simultaneous, both the characters are expressed simultaneously. When this F1 generation is crossed, in the F2 generation, it gives the ratio 1:2:1 ratio. So again this codominance which produces 1:2:1 ratio, but what's the difference? Here in incomplete dominance, F1 generation, half of the expression of dominant, but in this case codominance, both the heterozygote, both the parents are expressed simultaneously. Both the genes of the parents expressed simultaneously. So here half will express in incomplete dominance, here both expressed simultaneously. That's the difference among incomplete dominance and codominance. Then another example is lethal gene. So there is another example called lethal gene. This is observed in the year 1907 by E. Barr. E. Barr explained lethal gene in snapdragon. What's the other name of snapdragon? Antirrhinum. So in snapdragon, lethal gene is expressed, which is noticed by E. Barr in the year 1907. Let's see what is the example of this in the snapdragon. Here the example is snapdragon. In snapdragon, it produces 3 different types of plants. The first type of plant is green plant. Green plant, which consists of capital C, capital C. The allele present in this is capital C, capital C. Second type of flower is, so this green plant with chlorophyll pigment. We all know the plants which possess chlorophyll. Here another second type of plant is yellowish green. Yellowish green with carotene. So here the allele is capital C, small c. So it gives the color pale green. So it gives 3 different colors: pale green, golden color or aria, golden or aria. So aria color, it gives aria. The heterozygous condition, it gives aria. And the third is white color plant. So snapdragon, there are 3 different color of the plant. Green plant with chlorophyll, capital C, yellowish green with carotene pigment, pale green or golden color is otherwise called aria, and white color which gives the allele small c, small c. So what is lethal? Lethal is the condition in which the organisms which lead death of the organism. So lethal is the potential to lead death of the organism or the carrier individual. So this snapdragon which produces 3 different plant. In this homozygous 2, there are 2 homozygous, that is capital C, small c, 2 homozygous. This is homozygous dominant, this is homozygous recessive, and this is the condition heterozygous. So when this heterozygous capital C, small c, this capital C and small c is crossed with capital C, small c. So here the parent is capital C, small, the heterozygous parent. In the F1 generation it produces heterozygous. So this take it as a parent, and capital C, small c, capital C, small c. What is the color? Yellowish green, that is aria color. So when it is crossed, it gives the ratio 1:2:1, 1:2:1. In the ratio 1 capital C is to 2 capital C, small c is to 1 small c, small c. So it gives the green plant, 1 green plant and 2 yellowish green plant and 1 aria. This is the normal. But what happens here? This small c, the homozygous recessive. The homozygous recessive which causes death of the organism because it lacks chlorophyll. What is absent here? Chlorophyll pigment is absent. We all know if the chlorophyll pigment is not present in the plant, it can't able to synthesize its own food. It will die. So it causes death of the organism. So this 1:2:1 ratio is altered, and it gives 1:2 or 2:1 respectively. So this one already died. The plant is not present because it lacks the chlorophyll, it dies. it causes death of the organism called lethal gene. So, lethal gene which causes the death of the organism, the carrier individual which causes death of the organism. So, organism because of lacking of chlorophyll, it dies. So, it gives the ratio 1:2 or 2:1. This is the example lethal gene. Example snapdragon. Then pleiotropy. Another topic is known as pleiotropy. What does it mean, pleiotropy? The topic is pleiotropy. The single gene affects multiple traits. So, what is pleiotropy? A single gene affects multiple traits. Here there is an example when Mendel noticed it when he is conducting experiments in pea plants, garden pea, that is Pisum sativum. He observed this pleiotropy in Pisum sativum. So, what is pleiotropy? A single gene affects multiple traits. Single gene affects multiple traits. So, here the single gene which affects a multiple trait. Here is an example, the color of the flower. Actually, in Pisum sativum there are two different colors, purple color and white color. Then seed color, brown seed and light colored seed, and the dark spot in the axil of the leaf. So, these three characters, here it is one gene, a single gene which affects multiple traits. Let us see one example. Here the color of the flower, purple color and white color. There is a cross between purple color flower and white color. So, there is three different genes. The traits are three: purple flower, brown color seed, color of the seed brown, color of the flower purple, and the axil of the leaf which is dark spot on the axil of the leaf. So, this is one character, the three different traits together crossed with white flower, light colored seed and no spot on the axil of the leaf. So, these all together expressed. So, a single gene which affects multiple traits. So, this character is called as pleiotropy. So, these all comes under intragenic or allelic interaction. So, now the question is intragenic interaction. What are the examples? Incomplete dominance, codominance, lethal gene and pleiotropy. There are 4 different examples. So, it should be clear to you. The incomplete dominance, codominance, lethal. So, 4 different topics you learned today. Incomplete dominance, the molecular explanation. Learn all these 4 questions very clearly. Incomplete dominance, the molecular explanation. Codominance, what's the example? Then lethal gene, what's the example? And pleiotropy. What's the difference among incomplete dominance and codominance? In incomplete dominance, there are this half of this in F1 generation, the half R1, R2, only half will express in the F2 generation. 50% which express in F1 generation. These are the parent. In the F1 generation, it gives the color pink. But codominance, simultaneous expression. There is a simultaneous expression of the genes. Codominance. What are the example? Here protein chromatography comes under this cotton, Gossypium hirsutum and Gossypium streptianum is the example. So, it gives 1:2:1 ratio. Then lethal gene, example snapdragon. Snapdragon, it causes death of the organism because it lacks chlorophyll. There are 3 different types of plants. They are green plant which has the gene allele which contains capital C homozygous, and second plant color is yellowish green or aria. This example, this is one word question, aria, which is golden color, capital C small c, heterozygous, and white color is small c. Finally, pleiotropy, a single gene affects multiple traits. Learn all these questions very clear. Thank you.